Hello, everyone. Thanks for giving me the chance to tell you about some of my work. I hope you can see it in person at the conference in Montreal in just a few weeks. But the title of my talk today is Characterizing the Effects of Multi-Echo and Multiband Imaging on Corticostriatal Responses to Reward. So a little bit of a disclaimer, this is a little bit still in progress. There might be a little bit some differences between now and when I present at the conference, but I hope to get your feedback and improve it. So that'll be what we talk about at the conference. So, but again, I'm David Smith. I'm a professor at Temple University in Philadelphia. If you have any questions, comments, just feel free to email me here. Before I go too much further, I am obligated to say that I do not have any disclosures to provide. And also, while I have a little bit of a diversion, I want to go ahead and just take a moment to thank the members of my laboratory, my collaborators, my funding, and I also want to call specific attention to a few of the trainees in my lab who helped lead a lot of the work that I'm going to talk to you today about. So that was Jeff Dennison, Jimmy Weingarten, and also Ori Zaf. So these folks have been really helpful throughout this process. So it's been really fun working with them, like all my members of my lab. But if you want to read more about my lab and some of the stuff we do, you are welcome to check out our lab website. We will be recruiting PhD students for fall 2024. The deadline for that is December 1st. So feel free to check that out later and reach out to me. Okay, so a little bit of background. Our lab, like a lot of folks here, looks at reward processing and decision making. We know a lot of that circuitry, so that kind of evokes activation in the striatum, but we're not limited to the striatum in these sorts of studies. We tend to also look at some of the regions that the striatum is connected to, or that kind of connect with the striatum. During reward processing, whether it's social reward processing, non-social reward processing, all these different situations, we know the striatum is interacting with these different, so connectivity is very important. One example of this is one recent study that I did with my colleague. We found that connectivity between the default mode network and the striatum was enhanced during reward processing, just kind of underscoring that relationship between the striatum and other parts of the brain as a function of reward processing. But all throughout these studies and just throughout the field, I think there's always been this continual push to create better statistical power, whether it's through achieve better temporal spatial resolutions with the idea that maybe we can kind of boost the signal that we're getting, kind of get a more fine-grained understanding of how the striatum contributes to the reward, get a more fine-grained understanding of those connectivity patterns. All of those can be facilitated by better temporal and spatial resolution. What has occurred over the past 10 years has been multi-band or simultaneous multi-slice imaging, which allows for improved temporal and spatial resolution. Basically, we're in that approach, you're collecting multiple slices simultaneously, hence the name multi-slice. However, as with many things, there's always important trade-offs with these different decisions. So that's kind of the big focus of what I'm going to talk about today. That was motivated in part by just this larger question of like, what are the effects of the multiband on the striatal response to reward or just a signal in general? So if you go way back to 2013 and look at some of the tSNR maps, the temporal signal to noise ratio maps for the Human Connectome project, which used a multiband factor of 8, which is really high. That I don't think I've really seen higher than that actually, but this is really high. They're getting 8 slices simultaneously. They're working with a TR of about 720 milliseconds. One thing you can notice immediately is that there's a huge drop off in temporal signal to noise ratio down in the striatum, which kind of begs the question of how does that impact our ability to detect reward-related responses down in that region? So yeah, we were able to go faster in this study, but did we do so by sacrificing some of those responses in the region? You know, in my lab, we care about connectivity, so we want to kind of make sure we are balancing those different regions and not just kind of focusing on cortex or subcortex or vice versa. More recently, Brian Knutson and his lab have led this very important meta-analysis of reward processing studies that either use single slice, single band studies, so traditional fMRI, and they were able to compare the striatal response to reward to multi-slice studies. And one of the things they found is that the average effect size for those multi-slice studies was much lower than the single-slice studies. They followed this up with a nice empirical demonstration of this same sort of effect. And you can see that as a function, so as multiband increased, you can see a concomitant decrease in the activation in the striatum. And so they concluded from these results, from their meta-analysis and also their empirical work, that multiband compromises the detection of reward responses in the striatum. So one of the big questions I want to look at today is, are these decrements that are observed with collecting are these decrements that we observe, do we still see those when we're collecting multiple echoes, things that can improve the signal? So is there a way to kind of bring back some of the signals? So that's kind of one of the big goals of this study. So to look at this, we tested 6 different sequences. So we looked at multiband 1, 3, and 6. We looked at single echo versus multi-echo. We used 4 different echoes, 13, 31, 49, 67 milliseconds. We use a smaller flip angle than you might traditionally see. This is much smaller than the Ernst angle for all of the different TRs that we used. And this was motivated by this older study that showed that you're not really losing tSNR when you drop the flip angle that low, but you are able to reduce some of the effects of other artifacts. So we're kind of motivated to go with that smaller flip angle for that reason. We collected whole brain data for all of our 6 sequences with identical spatial resolution. The TR repetition time varied from 626 milliseconds to 4,700 milliseconds. But the scan duration and the number of trials were also matched. So we're not kind of giving one sequence an unfair advantage over another one, just by virtue of getting more trials. Same amount of trials, same duration. Naturally, the scans with the shorter TR will have more measurements, but that's one of the advantages of using those sequences. We also tested the 64-channel head coil and also a 20-channel head coil. I'll focus mostly on the 64-channel head coil today. There are some interesting differences I can tell you about at the end. Very quick overview. We found that the multi-echo is associated with higher tSNR and less signal dropout. Zooming in on the striatum for this effect, we found that so the multi-echo is denoted in the orange color and the single echo is denoted in blue. So what you can see from this plot of tSNR is that for both the 64-channel and 20-channel, there's no real differences in the, there's no real loss in tSNR for the single echo images. So we can crank up the multiband factor for those and not really see a big loss. But if we're using the multi-echo images, we do see a boost in the tSNR in the striatum. However, that boost is kind of much less for the higher multiband factor. So overall, the multi-echo gives us a greater tSNR in the striatum, but that is a little bit less with the multiband, the higher multiband factors. We did this in the context of a reward processing task. I'm not going to go through the details of the task. I just want to kind of focus on the basic gist. But basically in this task, people were faced with a decision, basically a gambling and guessing game where you have to guess whether or not the number on a card was above or below the number 5. If you guess correctly, you get $1. If you guess incorrectly, you lose $1. So we're focused mostly on this outcome period. Those are the results I want to kind of focus on. This is a classic reward task that's been used in a lot of other studies that was developed by Mauricio Delgado and used by many folks. So looking at that reward relative to punishment response, just looking at the question of like, are there sequence effects on reward activation? We don't find like huge effects on the striatal activation. We certainly don't see like a clear decrease in the reward-related responses as a function of multiband factor. If anything, we tend to see an increase in the reward-related responses in the striatum as a function for the multi-echo, particularly for the lower multiband. But this is kind of a reassuring result. We can say that we can use these more advanced sequences and not really observe any sort of loss in the striatum. We also looked at functional connectivity. So starting with the seed region in the ventral striatum and assessing a target region of interest that was identified from the contrast of reward greater than punishment, focusing just on the ventromedial prefrontal cortex. What we found here across both head coils is that the multi-echo, again denoted in orange, increased our detection power of that VMPFC VS functional connectivity. However, that detection power was somewhat reduced as a function, or somewhat attenuated with the larger multiband factors. So we are getting something from the multi-echo, and we're not losing a ton from the multiband factors. So summary, next steps. So, as with everything, there's always these important trade-offs with fMRI. So we're not really kind of prepared to say that the multiband is just completely compromising the striatal response. There's a much more nuanced picture. Of course, this is just a single slice through a very high dimensional space. So there's a lot of other work that needs to be done. One thing that we're really excited to look at in the future, hopefully before the conference, is how these newer echo-dependent or T-dependent denoises approaches impact these results. So that's something we've not looked at yet, but that's something we are in the process of kind of getting together. By the end of the year, we hope to have all of our data up on Open Neuro, some of our statistical maps on NeuroVault, but be on the lookout for that, and hopefully I have more to share on that front at the conference. So just again, I want to kind of thank my lab and my collaborators, and also our funding. So thank you very much. I'm going to stop screen sharing. Stop